From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on July 17th, 2024. Hello, Micronauts, and welcome to our Quality Quorum for today. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 50th episode of Matters Microbial. I am grateful to the podfather, Vincent Racaniello. I am grateful to the team at Microbe TV. I am grateful to Jennifer Quinn for all the graphical assistance. I am grateful to David Renata for editing. And I am so grateful to each and every guest on this podcast, and especially to each of you who listen and or watch this podcast. The folks at Microbe TV have mementos of this 50th episode. Please visit their website for more information if you're interested. As many of you know, Dr. Jennifer Quinn and I have collaborated on LuxArt, painting with luminous bacteria on nutrient media. Here is a wonderful piece Jenny did of one of the OGs of microbiology, Louis Pasteur. Please keep in mind that first-year college students can do remarkable LuxArt. Please have a look at this creativity from two of my students. It is always fun for Jenny and yours truly to be invited to institutions to put on LuxArt sessions. I am always so impressed with the creativity shown, and it helped defeat some of the negativity I hear about the microbial world in general. I also wanted to share the 3 laws of research that I came up with years ago and share with my Doc Martian research students. Rule number 1, 1 ego per lab, and it's not yours. Actually, your research belongs to the whole lab and everyone else. Ego is always the enemy. Rule number 2, sometimes research appears to resist investigation. Everyone has to fight through it to get that data. And rule number 3, the and research. It's not real unless it can be repeated. And each of these lessons were painfully learned by yours truly, and I hope that this plaque helps my own students. Turning to today's topic, if the majority of living things on Earth are unicellular and microbial, why does multicellular life exist? Now, this is a great philosophical topic, but it's interesting, wildly interesting to discover that fascinating research has been done to explore this very question. And one of the most visible investigators of this topic is Dr. Will Ratcliff of the Georgia Institute of Technology. As you're going to learn, his work is accessible, fascinating, and beautiful. Will, I have followed your work from afar, and it's an honor to meet you. Welcome to the Quality Quorum.
Thank you so much, Mark. I feel the same way. I've long admired your scientific outreach and just enthusiasm for all things microbial, and it's great to be here.
Well, it's good to have you here. And I think you have an interesting background. Could you briefly tell our listeners and viewers how you came to this? Because I was looking at your background.
Yeah, I may be one of the few people that works on microbes that got their start in plants. So my undergraduate degree from UC Davis was in plant biology. And I sort of got into that because I loved plants and was kind of a naturalist and would spend a lot of my summers hiking around our family property in Northern California in Mendocino County. And then I started doing plant biology, and I realized that plant biology was really like the microbial cell biology, the molecular cell biology of plants, which, while cool, wasn't really my interest at the time. And so I did a PhD with Ford Dennison at the University of Minnesota on plant bacterial symbiosis, because I was really interested in sort of studying ecology and evolution with still a plant component. And, I was growing plants in greenhouses, inoculating them with these soil bacteria, which form nodules. The plant makes a nodule that they live in. They fix nitrogen, which gives the plant critical resources to grow. And my plants kept dying and getting somehow infected by, like aphids in the middle of winter in Minnesota, which I don't know how that's possible. We're in an isolated building, but it's possible. And I got really frustrated dealing with slow, difficult plants, and I just started studying the bacteria. And I think that was the turning point, that I was like, oh, the microbial dark side. These things are awesome, very powerful, and fast. You could do an experiment in a matter of weeks, not a matter of months. It's wonderful.
By the way, my PhD was working with alfalfa and
Ah,
Rhizobium melillotae.
there
So,
you
and
go.
what were you working with?
I was working with Cynorhizobium melilota as well.
Yeah, I,
It's really
I
cool.
am. I developed a transducing phage for that particular bud.
Ah, that's cool. That's awesome.
Hmm.
Very cool. Small world.
Literally, literally.
Yeah.
And then this took you
Yeah.
to this interesting larger idea because it sounds to me like you've always had larger questions involved. And
That's
I want to
very
make a
true.
point of this if I can for just a second is
Yeah.
that so much of us in the past in particular have been reductive and we want to
Mm,
dissect things and
mm,
that's valuable in a lot of
mm,
ways. But
mm.
now we have the ability to look at larger questions.
Yeah. Yeah, that's a really good point. I've always, as a scientist, been motivated by questions and excited by questions. And I did a number of different things during my PhD. So I did plant bacterial experiments. I did some simple mathematical modeling on the evolution of aging, which was just spurred by random ideas. I worked on bet hedging and did some theory and did some experiments. And so I did a number of diverse things. Um, but the main thing that I thought about a lot was sort of social evolutionary theory and experiments, understanding how organisms evolve to interact with one another, under what conditions they will cooperate and maybe even build something cool, under what conditions that cooperation falls apart and the whole thing collapses. And it wasn't that far then to think that, like, okay, like we can understand symbiosis through this lens, two things which have sort of distinct evolutionary histories interacting together, maybe a plant and a bacterium. Multicellularity is another version of this kind of social dynamic, but it's one lineage which has then just gone off the wall, started simple, form a simple group of cells, and then ramped up the complexity of that group, evolving into the kinds of organisms that we see today. Plants, animals, mushrooms, seaweeds, modal algae. There's a lot of really cool multicellular life out there. And to your point about reductionism, it's a hard problem to study, partly because reductionist approaches don't tell you that much about origins. In a sense, like if I gave anyone here the space shuttle and said, this thing can fly, so work your way backwards to how like the Wright brothers first flew 100 feet. It would be really difficult, if not impossible, to try to work back the logic of taking something simple, getting emergent behavior out of it, bootstrapping that emergent behavior through either evolution or, in the case of the Wright brothers, engineering, making things better, iteratively improving. And the way that biology sort of is so good at evolving extremely fancy, bespoke regulatory mechanism that makes things robust, that makes things work, it ends up, changing the way that things work a lot from origins, where things are messy, things are emergent. They just kind of work at a multicellular level without having evolved for that purpose. But you have a lever in which natural selection can come in and can grab it and can sort of move the needle. And so realizing that sort of there's an opportunity to think about not backwards from the sort of modern era of the the fighter jets of the multicellular world, like animals, right? Something very, very complicated, but forwards, prospectively explore how a single cell evolves to form a group, and then how that group gains the capacity to have Darwinian dynamics at the multicellular level. How do they have a life cycle? How do they get new multicellular traits that are heritable? How does selection act on those multicellular traits and allow for open-ended Darwinian evolution of new multicellular traits, which we kind of know. We know that that has to be the way in which you take something which is small and simple and get something complex out of it hundreds of millions of generations later. But we still don't know a lot about those intermediate steps, how difficult it is, what you need, and how it in fact can progress. And so about 15 years ago, while I was still a graduate student, I began exploring these things with Mike Travisano, who's a professor at the University of Minnesota. Wasn't my advisor, but was someone I interacted with a lot and had a really great relationship with, and would just talk science all the time. And we were like, why isn't anyone, we should be doing this. And so then we started playing around with yeast, and it worked really well. So I've been working on it ever since.
This is where I think it's really good to take a moment and talk a little bit with folks about what has been commonly called snowflake yeast.
Yep. Yeah. So we started with a single cell baker's yeast, which is basically a pragmatic choice, and that it's a model organism in the lab that we know how to work with quite well. There's a lot of tools that are available for genetic engineering, synthetic biology, for ordering, every possible knockout, single or even pair of knockouts that's been made. Those collections exist. So there's just a ton of tools. And we thought, if we can get it working in this system, it's a good system because we can do a lot. We have a lot of power. And we have actually worked with other model systems since then, and they've been much harder. So I think it was actually a wise choice to use a model system that has a lot of tools available. And our first experiments were really simple. Like, we needed a way to get them to form groups, because that's the first step in evolving multicellularity, is get a group, and those groups then sort of take on the properties of Darwinian evolution, which would be reproduction, heritable variation, which affects future representation of the population, which we like to call fitness. That can either be because it increases growth rates and reproduction, or it decreases death rates and improves survival. Biology is a birth-death process, so all traits have to come back down onto one of those two things. And so we know that we need to get there, so forming groups is the first step. We did some experiments that didn't work out so well, but the thing that worked really well was just growing our yeast in test tubes of liquid media that's shaken, and then once a day putting them through a race to the bottom of a test tube. We would pull out a subsample, about 10% of the population, and then let them sink on the bench for just 5 minutes. And the things which got to the bottom survived. They went to the next flask. Everything else is thrown away. So if you figure out how to get to the bottom first, you win. And the way that they did that was evolving to form groups. And we called them snowflake yeast for two reasons. First of all, we did these experiments in Minnesota in December, and so when we first saw them actually like sinking through these tubes, it really looked a lot like what we were seeing outside of our windows, these big heavy snow clots that were falling down. But second, when you actually look at the structure of a snowflake yeast, and if you want to look at the PowerPoint that I provided, something like slide 8 would be a good example of this. You can see that they have this beautiful branched fractal morphology, which actually results from daughter cells not separating from mother cells. There's only one mutation that you need to take a single cell baker's yeast and turn it into a snowflake yeast. And the one which, there's actually a number of different mutations that you can do to mess with the cell cycle and break the process through which daughter cells basically break down the cell wall connection between a mother cell and a daughter cell. If you break that, you get daughter cells that don't separate, and so they grow sort of this branching, three-dimensional, for lack of a better word, they look sort of like a bush growing in 3 dimensions with branches coming off, where the first branch is big and the second branch is exactly half its size, the third branch is half its size, and they get this kind of nice fractal ordering.
Have you done kind of like I'm going to call this confocal, but I'm not using the term correctly, but have you gotten 3-dimensional images of these?
Yeah, we've gotten many. When they're small, it's very easy to do because you can use a confocal or a light sheet microscope and you can see every cell. And in fact, the images on slide 8 that I mentioned were a confocal. As they evolve to get bigger, so when the earliest snowflake yeast are on the order of 100 cells, before actually strain resulting from their own internal growth causes them to pop a branch off, and actually leads to a life cycle where you get babies popping off. And that turns out to be really important and cool. But over generations, we're selecting on them to essentially win the arms race to get to the bottom of the tube. It's not sufficient to be at the bottom. You have to beat everyone else. And over generations, they evolve to get a lot larger by evolving to form tougher bodies. And once they get big, and by big I mean like bigger than a fruit fly, millions of cells, then it becomes very, very difficult to image them in 3 dimensions, for the main reason that if light is going through a cell and then into open media and then through a cell and into open media over and over and over again, every time it does that, it diffracts a little bit. And so you can't see more than about 50 microns into a cluster that might have a 600 to 800 micron diameter. You can't see more than the outer shell of that thing before the light just gets so attenuated that it goes dark. So we had managed to see inside those guys through an extremely labor-intensive and expensive process of serial bulk-based scanning electron microscopy, where you basically take your cluster, you embed it with something that absorbs electrons like osmium, and then you embed that whole thing into a block of resin, and then you take 50 nanometer slices, and you do about several thousand of those, and just slice your way through it, and then image every single one with an electron microscope, and then recombine all of these stacks into a giant three-dimensional image, which is quite a bit harder than you might expect. We had a physics graduate student, spend about a year just working on reconstructing these images because the software solutions that we found didn't work so well. So we did the entire thing brute force in Python, creating three-dimensional images as just matrices of RGB. Actually, it's just matrices of grayscale 0 to 255 pixel density values. And so just making
Let,
images from, essentially a spreadsheet of brightness values for every single pixel.
so let me
But it
jump
worked.
in really quick. No, this is
Yeah.
beautiful work. Number one, some of these images are almost artistically beautiful.
Thank you.
And more than once, I have been asked, very much like you've probably seen the Panabacillus colonies, and people will
Yes,
say
yes.
things like, have they been manipulated in some ways? And
Mm,
probably some of those have, based on what I've tried anyway.
mm, mm.
But what from my understanding, there's been no manipulation of those beautiful images. So that is how they look to you.
Only staining to be able to see certain features.
Sure.
So I'll call out a few beautiful images if David wants to show them while I'm describing them. The one on slide 30 actually shows our ancestral snowflake yeast on the left, and from the tube level, looking at the test tube, an evolved one that's large on the right. You can see how big the groups evolved to be. And then you'll see this movie of a blue cube rotating, and that's just a little slice from the inner core of one of those large macroscopic snowflakes, just spinning it around, adding layer after layer after layer. You can almost see it has this slightly blocky texture because it's kind of being reconstructed almost like in Minecraft, where it has like these just definitive layers to it. Um, if you look at, let's see, slide, I've already talked about slide 8, which is confocal microscopy. And here, color is just depth from the viewer. So it's just this position of the Z-plane. We have others that I think are really beautiful, like slide 39, which is another kind of confocal microscopy. And here on the upper one, we've stained nuclei in orange and cell walls in blue. And so, in fact, if you look closely, if you can zoom in, David, it'd be really cool. You can even see some areas where the nucleus is in the process of moving from one cell to the other. And you can see others where the nuclei are actually sort of pushed apart, but there's a thin orange thread connecting the two. And that's the mitotic spindle. In yeast, they have this mechanism of mitosis where they actually have to, use mechanical power to push the nucleus from one cell to the other. And so the mitotic spindle sort of remains behind as these things are pushed apart.
So there are connections between the cells.
There are connections while they're dividing, but after
Got it.
they divide, you do see a septum form. And so they don't remain open the way that some filamentous multicellular fungi have just, these open connections from cell to cell. Our cells are firewalled off from one another.
And you got to admit they're artistic. I mean, I'm to the point where I want to get one framed for my wall because I think it's beautiful.
I would, if I could teleport you here to Georgia Tech, we have a whole gallery in our hallway outside the lab. A research scientist in our lab, Tony Bernetti, also thinks they're artistic and has made many, many transformations and images. I think I secretly think he did this because he knew they would be so pretty, like labeling different components because they would be fantastic to look at.
Sure.
And so we have, several, maybe 100, 150 images along the walls as you walk down the hallway, and they're really cool. Whenever I have visitors here, I take them there because I can describe the science best through the images. And I'm actually in the process of collaborating with an artist who works in wire to make a wire sculpture of snowflake yeast, which I think will really bring the sort of morphology of them to life. If you can see it, and sort of it's in 5,000 fold larger than real life, but 3 dimensions, not 2.
So for the listeners and viewers, I want to really emphasize the simplicity of the initial experiment, the complexity of what resulted. And there's no reason that anyone couldn't have started with this kind of thing. They might not have taken it as far as you have, But that's what I love about this work is that it starts with something that's relatively simple. Who falls to the bottom of the tube first? And every microbiologist has seen this. Micrococcus luteus is really
Mm,
tough to grow in tube culture
mm,
because it tends to fall too quickly.
mm.
Um, and so this is something that we've all seen, and we just kind of look at and move on. I did have one question for you, and I hope it
Yeah,
doesn't throw you off track. Now,
sure. Yeah.
you started with Saccharomyces cerevisiae, baker's
Mhm,
yeast.
mhm.
Does this also happen with wild yeasts?
We've never done experiments with wild yeast, but so I would expect that it would. There are we keep getting more and more, work sort of showing that wild yeast are not exclusively unicellular in the first place, right? There's like a varying amount of multicellularity in nature. You have things which form aggregative groups through flocculation, which we've known about for a very long time. And quite honestly, that's what I thought we would get when we started our experiments. Snowflake yeast are different than aggregates in a way that maybe we can talk about after this, and that turns out to be very important for their evolution.
Please.
Um,
Yeah.
but there's also yeast in nature that form clumps of cells through mother-daughter cell adhesion after division. So those clumps remain clonal if they started with a single cell or if they have bottlenecks when they fracture. They're not as big as snowflakes, and typically you don't see things that have like this characteristic snowflake morphology. They tend to be a little bit more like uh, they look more like a bunch of grapes with a little bit more of a disordered structure rather than this beautiful
Yeah.
sort of outwards growing branching structure.
It's like
Um,
Staphylococcus, like
yeah, 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 yeah.
Streptococci, yeah, or
Absolutely.
Streptococci are just a conga line of
Yep. Yep.
cells, right?
Yep.
And
Totally.
Staphylococcus form or a Sarcina for that matter, as you often see. So this clumping issue is one thing, and
Yep.
groups of
Yep.
cells is a second. But what you're
Right.
finding is this emergent property, which is fascinating.
There's a lot of emergent properties. I think, writ large, what we're finding is that emergent properties of multicellular groups give you a lot of behaviors and traits that we assumed needed to evolve for that purpose. But once you form a multicellular group, you get a lot of things just for free, and that they didn't evolve for this reason, but they provide a mechanism through which Darwinian dynamics can go and you can improve them because you can generate heritable variation in these traits, and if they improve fitness, then they can increase in frequency, and you can do this open-ended process of building upon what you had in the past.
And
So, yeah.
I
Oh
was
no,
going
go
to
ahead,
say
Mark.
one further thing for listeners and viewers. One of the really cool things about doing microbial science today is you can literally determine what genes have changed. So for example, if I were to do a similar experiment, and I have, where you look at the things that swim fastest,
Hmm.
and
Yep.
you end up with things that swim a lot faster than the original type strain, then you can get those genomes done, compare it to the type strain, and determine specifically what genes have changed for about $75.
It's
And
very
this is
expensive.
it is, and
Yeah.
that and we're so used to thinking of one gene at a time, one
Right.
phenotype at a time, and what we're
Right.
learning is all these things can now be looked at in groups, and this is so wonderful.
And I'll say one of the very nice things about long-term evolution experiments, and ours is now, I would consider it to be long-term. We're about 1,500 passages through this growth size selection regime, and I hope to do this for the rest of my career. Like
Mm.
I feel like I would be missing an opportunity with my life if I didn't push snowflake yeast as far as I could before I die or retire. So I'd like to continue doing this for several more decades. And this is one example of a long-term evolution experiment. The long-term evolution, the LTEE that Rich Lenski started with E. coli has been running since the late 1980s. It's gone over 75,000 generations. It's called the long-term evolution experiment, LTEE, and there's a number of others, right? There are field experiments, most famously perhaps Peter and Rosemary Grant's Darwin's Finches experiment, right? Long-term evolution experiments teach us a lot about evolutionary processes that you simply can't get from an experiment that's really short and only looks at a snapshot in time. But also, to your point, Mark, they actually have an element of future proofing embedded in them, and that if I have, every 100 generations, I've frozen all of my populations in a -80, then I can come back. I can, interrogate virtually any question that I have because I can pull out any future strain and its actual ancestors. I can sequence them. I can see what's different. I can reconstruct things. I can, test hypotheses in sort of almost any manipulative way you want. You know what environment they evolved in. You know who's related to whom. And if future technologies arise that are better than what we have now, then you can use them on past samples. So there is something very, I think, special about something where you can collect the entire experiment in sort of a time capsule, and anyone at any time can sort of thaw it out and use it and ask whatever questions that they have now. And I think often some of the biggest insights that we have from long-term experiments in evolutionary biology aren't those that we set out to sort of, ask, but are things which occurred through the process of doing the work, the experiment generating new exciting hypotheses, and then us testing those hypotheses with the experimental system, and then complementary techniques like mathematical modeling or synthetic biology.
And I do want you to continue, but one further thing I wanted to remind everyone is that I hope everyone appreciates the number of different kinds of people that have become involved with this.
Mm.
I do think that we tend to bin ourselves off into different groups. And then the problem with that is you don't get the added perspective. So I know you use a lot of mathematics in what you're doing. You're doing a lot of computer programming. You have
Mm-hmm.
physicists involved looking, and I
Yeah.
know you're studying flow dynamics, all
Yes.
kinds
Yes.
of things that come
Yeah.
together this way. And that's really key to modern science, is to bring in lots of different viewpoints.
I couldn't agree more. I mean, to me, I wouldn't. I don't know if I would love science if it was just me in my lab doing experiments. I think I would feel. I don't know. I mean, for me, collaborating with other scientists, getting their like insights, which are totally outside of what I would have come up with and are great ideas, having the opportunity to really spend a lot of time with scientific friends and like working with them on projects, like that adds a lot of joy to the process for me.
It does.
And I think, some of the most exciting results that we have are things that we would have never been able to do because they have to do with physics that are not trained as a physicist, but if you're evolving in multicellular body, a lot of what you need to understand are how the changes in the way that cells interact with one another both constrains the system, but also creates opportunities for things at the multicellular level. And we'd have hints that these things were important, but we would effectively just be blind to all this exciting science, right? And so, for me, collaborating has been both really scientifically enriching, but also very personally enriching. That it's great to have these. I have collaborations that have lasted for more than a decade, and I look forward to talking with my friends who are also my colleagues every time we meet.
It's wonderful. So you were telling us a little bit more about emergent properties with your snowflakes as they grow particularly large.
Sure. Yeah, yeah, yeah. So let me find a good slide because I think this is one that's illustrated very well from images. So if we go to slide 12, and this will play into slide 13, David. Um, in order for like a group of cells to become a Darwinian entity, it has to have the properties of a Darwinian system. That is, it has to be able to reproduce so that you have, essentially multiple copies of this thing in a population. And then you have to have a mechanism for generating variation between these replicating entities that's heritable, so it can be passed on to offspring. And those variants have to affect their frequency in the population. They have to affect their fitness. So we sort of know from a philosophical, from a theoretical sense, that in order for the transition to multicellularity to occur, and that term is loaded and means different things to different people, but I'm using it here to specifically describe a transition in evolutionary dynamics from natural selection acting on reproducing single cells and optimizing, increasing the fitness of individual cells to, going up a level to acting on the properties of reproducing groups of cells. All right. So we know that, groups have to grow. And if you play the video on this slide, David, you'll see that snowflake yeast have this kind of nice synchronous division pattern where they grow and divide and daughter cells remain attached to mother cells. And then they kind of, you can see it getting quite crowded as they fill up the interior of that space. And if this thing is growing in 3 dimensions, there's plenty of room on the outside, but on the inside it begins to get dense. They begin to run out of room, and cells continue dividing in that dense space. And what happens when you run out of room, but you keep pushing cells in through division, is that you get strain accumulating on these branches of cells, and eventually that strain exceeds the strength of a cell-cell connection. And much like cutting a branch off a tree in your yard, you cut a cell-cell connection and a branch falls off that snowflake yeast, pops off the cluster, and becomes a propagule. And it turns out that every time you break a branch, there's a cell at the bottom of that branch that is the mitotic parent of everything downstream. So you actually have genetic bottlenecks embedded in the structure of the snowflake yeast, not because they're good. Side note, they're really good for multicellular evolution, not because they're good, just as a side effect of geometry. And if you could imagine that if you have a snowflake cluster, and actually go forward 2 slides if you don't mind, David, and you'll see a light sheet image of a snowflake rotating in 3 dimensions. If you imagine that you have a mutation occurring somewhere in that cluster, and you have a whole branch of mutant cells, so this is now like there's 2 genotypes in one cluster. If I were to take that cluster and put it into fresh media and come back, 36 hours later, there would be a million clusters. Let's just turn off mutation for the case for this example. If there's no more mutation, I will have at most one chimeric cluster, one cluster that contains both genotypes. Because if you imagine where fracture can occur, it can occur between 2 mutant cells, giving you a mutant-only propagule. It can occur between 2 wild-type cells, giving you a wild-type-only propagule, or it can occur between the first wild-type cell and its mutant cell, giving you 2 clonal propagules. So there's basically no way to have genetically diverse groups without very, very high mutation rates, given the rate at which these bottlenecks are purging genetic variation. So we can think of snowflake yeast as being effectively clonal, where every cell is genetically identical. And this is the same as you, me, dogs, most trees, most mushrooms. Complex multicellular life is typically clonal. And there's a lot of really good reasons why clonality favors the evolution of increasing multicellular complexity. One reason is that it sort of prevents within, cell level selection within an organism from being a problem. In our case, this often does manifest as a problem because we're so big, 39 trillion cells, and we live so long that we have enough scope in our life for mutations to generate variation. And that can sometimes lead to within organism conflict, which we call cancer. But starting from a single clonal genotype sort of wipes that slate clean. You could imagine it'd be much worse if I started with a billion cell propagule that already contained genetic diversity. The second thing that clonality does is it allows selection acting on the properties of groups to drive Darwinian processes really cleanly. If you imagine that there was 50 different genotypes in a group and selection's acting on the trait of a group, well, now it's actually acting on, potentially a bunch of different genetic bases for that trait. And then if you reassemble those genotypes in different combinations, maybe you don't get this trait again, right? It sort of makes the heritability challenging. But if every group has just one genotype underlying it, because every cell in the group is clonally identical, then any group traits that they express have a pretty good chance of being, heritable. That is, if you create another clonal group with the same composition, it can grow into something that has the same traits. Another way of saying it is that you basically have this really nice identity between the traits of groups that might just be emergent properties of how the cells in that group behave, and the genotypes in that group that create that emergent behavior. So clonality is great. It solves levels of selection conflict issues. It also allows selection on emergent multicellular traits to be more heritable. And there's even good reasons why if you can basically partition all the genetic variation in a population into different groups, then selection acting on those groups changes the frequencies of genes that are related to multicellular traits as fast as possible. If I was selecting on a group that had many, many different genotypes, I can't really change genotype frequencies in that population very quickly. But if you bin everything, that creates big groups into here and small groups into here, and the big groups win, then you can just throw away small groups really fast. So it makes selection on multicellular traits maximally efficacious. So there's a lot of very good reasons that clonality is important for the transition to multicellularity. Snowflake yeast they have this from the get-go. There's one mutation that takes a single cell, makes it into a snowflake yeast. We use the mutation ACE2. It's a transcription factor which regulates daughter cell separation. You just knock that out, you get a snowflake yeast, and you have this beautiful fractal 3-dimensional growth form that has a life cycle. In fact, David, if you don't mind, going to, I have an overview slide for this material. Slide, I believe it's 27. I think I was reading the numbers wrong before. I'm sorry. I was reading the number above it, not below it. This is slide 27. And, you know, with a single mutation, we get this emergent multicellular group, which daughter cells just remain attached to mother cells. It breaks off that grows until it runs out of room, breaks off branches. So you have something which reproduces, right? One group will literally, if you put it in a flask of media and come back 24 hours later, there'll be thousands of them, and they'll all be the same size. Offspring from the first group, which themselves have had their own offspring, and you get multiple generations of snowflakes. And mutations within those snowflakes change the way that cells behave. They don't have developmental processes yet. They don't have like the kinds of development that animals and plants have, where you have genetic modules that sort of have evolved for the purpose of scaffolding the emergence of, sophisticated three-dimensional structures. They're just dumb clumps of cells that don't even know they're clumps of cells. But you get mutations that change the behavior of individual cells. For example, one of the main things we see is things which delay progression through the cell cycle, so you get more and more elongated cells. And if you make those cells longer and longer, that changes the way that cells in the group interact in a physical manner. So if I make my cells twice as long as the ancestor, they can actually grow a lot bigger before they end up running out of room in the interior and breaking branches. So you get bigger, tougher groups simply by changing a property of individual cells. There's no coordination, there's no regulation, it's just emergent physics. But you get much larger, much tougher groups, and it's really heritable. In fact, the emergent multicellular properties of snowflake yeast are more heritable than the same trait expressed in single cells, which is astonishing, but and there's good reasons for it that I don't feel like I need to go into the weeds for. And so they've got this they're a blank slate through which open-ended Darwinian evolution of groups can occur. We have something which creates groups, which creates a life cycle in which groups reproduce, which results in single-cell genetic bottlenecks for every group, so they're clonal, that allows mutations that change the properties of cells to segregate between groups, change the properties of groups, and now natural selection can see those properties of groups and it can act upon them. And when it's acting on those group properties, it's also acting on the underlying genetic basis for those groups, because every group is clonal. And you can imagine you just kind of keep cranking through that generation after generation, and you start stacking multicellular traits. And now you have the opportunity to undergo sort of process of open-ended multicellular Darwinian evolution. And at this point, our very simple little snowflake yeast is still a very simple little multicellular organism, arguably as simple as they come, but it's clearly on the path to evolving increased complexity. And that's what we're doing in our lab, is we're sort of running out the open-ended evolutionary nature of these, and we're seeing what we can get.
It's fascinating. And I, even physically, I would think increased agitation would lead to changes in the morphology of what you would get. And those two would be heritable in a different way by accumulating mutations. And this is really interesting because I was thinking a little bit about when you mentioned cells going into business for themselves. And we see this idea of cheating when you talk about ecology, where some members of a group take advantage of a group behavior. Same thing's true for mat formation in biofilms, mixococcus with spore formation. And what's interesting to me, and this brings me back to the first question, is why does multicellularity exist? Because one could make the point that being a single cell might have an advantage over being a large group of cells.
I think it usually does, and that's why multicellularity is only evolved. I don't know. We know of 50 transitions that we like to say it's evolved 50 times. I think in bacteria and archaea, it's way more prevalent than that. We just don't typically count them for bad reasons, that they're different from eukaryotes, and so we don't really count them, even though they have their own way of being multicellular. But still, most lineages don't evolve multicellularity because most of the time it doesn't make sense. And there's no one reason why multicellularity makes sense. There's a bunch of different reasons, and it really depends on the ecology of the organism. But multicellularity typically only evolves when there are benefits associated with increased size. So when we're talking early, the transition from a single cell to a simple multicellular organism would be driven by something like the benefits you get from being a bit diffusionally separated from your environment. protect, right? Because toxins can't get into all of your cells, or
Right.
predators can't eat you because their filter feeders in their mouth is too small. And if you form a group, then you're immune from predation from that organism, right? These are things which actually will drive the evolution of simple multicellularity. You can see benefits to collective metabolism. So, like if you're Vibrio and you're trying to break down some poor dead shrimp exoskeleton, right? A biofilm is going to do it a lot more efficiently than one cell, where any glucose that's liberated just diffuses away, right? But if you're sitting there with a couple billion of your friends, you can build up a pretty high concentration of, whatever it's called, chitinase. Break it down and eat it. So these kinds of sort of concentration-dependent, metabolic functions probably often do benefit from having a high concentration of organisms in one space, and multicellularity helps you get there. And if you can do it clonally with a bunch of genetically identical cells that are also contributing to this, you might be able to avoid some of the problems of social conflict arising from cheats. Motility is another reason to become multicellular. That often there are physical scaling laws that favor forming, a bigger group of swimming cells over a single cell. And so, or, even physical motility like slime molds that will, go from being single cell amoebae into these actually motile slugs, which will then work their way through the soil and find a good place to sporulate. So for simple multicellularity, the benefits, they always have to outweigh the costs, or you just won't have it, right? But I think, and we shouldn't look to one benefit from multicellularity. The benefit really stems from size, and under what ecology does forming a large group of cells provide a benefit?
That's
And then,
fascinating.
yeah, once you have that, now you have the opportunity to sort of begin evolving to do things that single cells just can't do, right? So the complex multicellular organisms, plants, animals, mushroom-forming fungi, seaweeds, they have created their own ecosystems based on things that they can do that single cells aren't very good at. Single cells don't form large, motile, heterotrophic predators that run around and chomp things. They can do it at the single cell level, but they're not, but like animals can do it in a way that nothing else can. They've occupied this niche of large, mobile, heterotrophic predators. Plants, they're fantastic at growing outside of the water. Their ancestors are aquatic, right? It's not invading land wasn't the hard part. Getting out of the water was the hard part. If you're an underwater, alga. But they've solved water and nutrient transport problems. They've solved mechanical problems of forming big, tough, bodies that can withstand going hundreds of feet in the air. And so they're doing things that single cells can't do because they are now evolving into multicellular niches through key innovations, which sort of allow adaptive radiation and expansion into a kind of an open new niche space.
You've really opened my mind to a lot of things. I mean, I had heard the concept, and I think you have a video that illustrates this, that one possible advantage of size is to avoid predation. So you have a
Mm-hmm.
protistan that
Mm-hmm.
can feed on the individual yeast, but can't literally fit snowflake yeast
That's true.
into its I guess you can still call it a mouth. I have questions about protists because they're very unusual. Um,
Yes.
but I love that idea a lot. But nowadays, and I don't know if you have had a chance to look at this, but Roland Hotzenpiekler, I had to make sure I can
Yep.
pronounce that properly. He's a
Yep.
very wonderful person we've had on the show. Recently
Excellent.
put out a paper about multicellular magnetotactic bacteria,
Yep.
suggesting that there might be a division of labor and some heterogeneity within the group.
Yes. I wrote a primer on that paper. I really like that paper. Yeah. So they used nanosims and Bondcat, pretty fancy techniques to look for spatial metabolic heterogeneity amongst these cells and these multicellular magnetotactic bacteria, and it really does look like there is the potential for between cell metabolic differentiation. We don't know that that means division of labor yet. We don't know what they're actually making, and we don't know whether or not they're exchanging it through the sort of it looks like they're exchanging it through the acellular space in the center of this thing, but they don't know that for sure. But this is a fantastic organism. I really think that bacteria and archaea get sort of short shrift when it comes to multicellularity, just because we look at something like a tree, we go, oh, look at that. There's a lot of cells and they're spatially differentiated. That's multicellular. Bacteria and archaea are doing it a different way. They're often temporally heterogeneous. They're often cryptically multicellular in a way that if you don't have fancy high-powered techniques, you don't see the differentiation because it's not obvious. It's not obvious
And,
till you get in there and see what the heck they're making and how they're interacting with each other, right?
and since I've spent my career looking at what I call undomesticated microbes, one of the
Mm,
really interesting things
mm,
is that undomesticated versions of the lab rats that we grow in the
mm,
lab
mm,
often associate with one another very differently than they do after they've been selected for to live in the
mm,
lab. I have a saying that the hand of Darwin is on all that lives. And I also call it Darwinowing. And we
mm,
forget that we're Darwinowing in the lab, just like you're
mm,
doing with snowflake yeast.
for sure.
But this is what we do. We take the bacteria that grow most efficiently on the media that we use, the most quickly, et cetera, et cetera. And so they don't necessarily reflect what's in nature. So if I took trees from outside and I tried to grow them just as fast as I could and did that for thousands and thousands of generations, I would
Right.
get things fundamentally different than the tree outside.
You would. You would probably get back to having some little leafy forbs,
Yep.
which interestingly enough, trees have evolved many times. They are not monophyletic. It's interesting how easy it is to evolve to be a tree, basically,
So
have
so
secondary growth as a plant.
so this is fantastic work. What kinds of things are you going to look at in the future
Yeah.
with these snowflake yeast? And it's
Yeah,
easy to say, I'm just going to see what happens because that may be part of it.
that's part of it. Yeah. So I guess we have like two major branches to our lab. One is running the multicellularity long-term evolution experiment, which in homage to Rich Lenski's LTEE is the M lowercase u LTEE, which the acronym is multi. So we had to do it. So we're running the multi, and that's really run by Ozan Bozdog, a research scientist in my lab, who kind of came up with the original selective scheme and has really been the master of the entire experiment, propagating it. He comes in 7 days a week and has for 8 years to run this experiment. Without him, I don't think it would exist. And I'll tell you more about that briefly, like sort of where it's gone and where we think it's going. And then we also do sort of more specific hypothesis testing with synthetic biology and mathematical modeling, right? So the kinds of experiments we do there don't involve tons of evolution and kind of seeing what happens and then deciphering it through experiments, but more like we think this is important. Let's make a strain that has the following behaviors and then see if it is, right? So, for example, there we recently engineered snowflake yeast to express oxygen binding proteins. So, myoglobins from a sperm whale. We yeast optimized it, put it into yeast. They grow like meat colored, they're like pink. Or peanut worm, myohemerythrin, another oxygen binding molecule. Because for them, the thing that really limits them is getting oxygen to the interior.
Ah, okay.
And so we're like, well, what do animals do? They make things that bind oxygen, and then we can make beautiful three-dimensional biophysical models, and we can even evolve those strains. And yeah, of course, it works, it helps, and there's some cool unexpected stuff that I don't have time to unpack there. Um, One of the things that has long been and a hobby horse is too strong of a word, but like I've long been frustrated that in evolutionary biology, specifically thinking about multicellularity, we don't disentangle things that are obligately multicellular. So like we are an example of one that's obligately we have a single cell phase that's completely transient that just goes right back to multicellular groups. From things that are clonal. So like there's lots of obligately multicellular organisms and lots of facultatively multicellular organisms like slime molds that will transition between single cell stages and group stages. Um, and so we kind of have this neat division that like everything that's obligate, except for these multicellular magnetotactic bacteria now from Roland and others, everything except for them that's obligate is also clonal. And then the facultative things are a mix, but most of them are not clonal. And so we've kind of confounded clonality and the consequences of that in terms of the evolution of the system with whether or not it's obligately multicellular or has a persistent single cell stage. And so we've engineered snowflake yeast to basically alternate between growing as single cells and growing as snowflakes. And so then we can actually evolve them and compare that evolutionary trajectory to things that are obligately multi and obligately uni. And it turns out that having even a short single cell stage completely changes their evolution and just pins them onto being like single cells. Like it, and for reasons that we think we understand pretty well. So it turns out that that's really important. That's a hypothesis-driven type of experiment. And we can do that with synthetic biology, engineering, and experiments, and models too. But in the multi, that's more open-ended, right? We're letting that thing run out. And so I'll just tell you a few of the greatest hits, things that are pretty cool, things that have already happened and things that are in the works. So they evolve over the first 600 transfers, which is about 3,000 generations, to go from being microscopic 100 cells to macroscopic like 500,000 to 1,000,000 cells. And the toughness of the organisms goes from being 100 times weaker than Jello, very brittle. A snowflake yeast at time 0 is a kind of terrible. I told you, I extolled the virtues of snowflakes and how they have this life cycle and all that. But they're also, as a material, they're very weak. They're only one cell-cell bond away from breaking apart, which is actually worse than most materials where cells are connected to everyone around them and they kind of act like a bulk material. They're not a bulk material. They're a branchy material, which is very, very flimsy. And so we're selecting on them to get big and to get strong. And what they evolve to do is they evolve a clever trick. The cells get longer. The connections between cells get about two and a half times bigger. So they get a lot bigger and they have a lot more material in there to hold on to cells. And when they get those two things, they're able to stay together long enough that the cells begin to wrap around one another before the group breaks.
There we go.
And once they entangle like that, they actually become really strong, as strong and tough as wood. So they go from being terrible material to a fantastic material. And that allows them to survive the strain required to get big. So that's pretty cool. Now you might imagine that getting big results in crazy resource limitations, right? Diffusion limitation. Anything that out there that's multicellular that's big has some way of ameliorating resource limitation. They have a circulatory system, or they're like a sponge and they push water through their tissues. To our surprise, even if we grow these snowflake yeast, they're growing normally in shaking incubation. Even if we grow them in static media, where diffusion limitation should be very strong, you should run out of. Our physics colleagues are like, they should, we can calculate this. They should run out of glucose after 50 microns, maybe 100 max. These things have a diameter of 200, 300, 400 microns, and they're still growing exponentially, meaning they are not slowing down their growth rate at all as they get bigger. How are they doing that? Well, David, you can show a video on slide 48. They actually create emergent, trying to show this on my thing. They create emergent fluid flows where they are big enough that as they metabolize glucose in the interior, it actually becomes a less dense fluid in that interior space because they're making ethanol, they're consuming glucose, and it becomes less dense. And so denser media from the outside pushes through their porous bodies. Their bodies are only about a third cells, and the rest is just kind of open web space, right? So they're like kind of like a sponge at your sink. You can push water through. It pushes through and then pushes the less dense media up and out the top. And if you go to the next image there, you'll actually see there's a volcano-like flow shooting out of the top. These things are flowing media at really high rates, hundreds of microns per second. They're flowing media at about the speed. It's about like their radius per second. And we think that, well, actually we have strong evidence that this basically overcomes diffusion limitation and allows them to essentially mix their environment themselves so they don't run out of food, even though they're really big. And this allows them to grow exponentially to ridiculously large sizes. We also have evidence that they're beginning to evolve cell specialization. So we've done single cell RNA sequencing of our yeast. And when we do that, over time, we can see like kind of initially they're all kind of just they're all doing kind of one behavior in sort of, gene expression space, for lack of a better word. But then over time, you get a second group and then a third group popping up. And those things are overexpressing different kinds of genes. So we sort of, this is not published work. This is future work. It's work we're working on right now. But we think the sort of first cell group are just normal happy cells that are growing and metabolizing. That second group that pops up around 1,000 generations in, they're overexpressing cell wall biogenesis genes. So we think they're actually maybe making a subset of cells that are tougher more able to withstand mechanical strain, which would be really cool in a spatial context. Especially if the older cells that have lots of branches are also making more cell wall. And then that third group looks like they're turning on genes for programmed cell death or cells committing suicide. And this isn't a trivial number of cells. We're talking 20% of cells appear to be doing this. And we actually think that this is their way of re-engineering a life cycle. That these entangled yeast, remember before how I've extolled the need for a reproductive life cycle, that groups have to grow and create offspring. In our experiment, I didn't mention this, but we create a very strong incentive for groups to reproduce and make offspring and not just grow forever. Because if we do that, then the whole population becomes one group. And we're no longer really exploring multicellularity, we're exploring biofilms or something like that. But you'd have only within group evolution. You wouldn't have between group evolution. We need our things to be discrete and competing against each other so we can select on groups. That's essential to the whole process. And so, we throw away about 90% of our population before even doing the race to the bottom of the test tube. And that means that even if something had like perfect survival during the race, it only has a 1 in 10 chance of even getting to race in the first place. And if they don't make 10 offspring, then they get, they're just going to get diluted away to zero. So there's a strong requirement that we actually dilute our populations by about 32-fold per day. You make about 32 offspring a day, otherwise your numbers should just attenuate over time. So there's a requirement based on the way we set up our experiment that groups create new groups. Entanglement gives you wild mechanical toughness. It gives you large size. That helps you win the arms race to the bottom of the test tube. But it comes at the cost of a reproductive life cycle. If you're really good at staying together, then you don't make any offspring. So after entanglement evolves, that's when it looks like they're actually evolving programmed cell death, potentially as a mechanism of breaking connections between cells and shedding little branches that are not entangled. And in fact, at that time, we see that the profile of propagules going from being tens of thousands of cells to there's a lot of now 5-cell propagules, things we think that are on the periphery that are broken off. I got 2 more things I'll talk about really briefly, if that's cool, Mike.
Sure.
Just future stuff. All right, so our snowflake yeast also undergo a whole genome duplication and become tetraploid. Yeast become tetraploid fairly easily, but it's unstable, and typically you see it sort of regress back to being diploid over maybe a year of in-lab evolution. Lots of experiments, and the typical thing is for them to regress back and just lose copies of those chromosomes. Our yeast become tetraploid super early, 50 transfers in to an experiment that's on 1,500 transfers now. 50 transfers in, they are tetraploid, and they stay tetraploid the entire time. There's really no losses. And they become tetraploid because tetraploidy gives you bigger cells that are more elongate, that give you bigger groups, and so it's essentially under positive selection this entire time. And then in our populations that evolve large size, which is actually not all of our treatments. I didn't talk about this, but we have 3 treatments in the multi, and only one evolves microscopic size. In that one, they're actually leveraging aneuploidy. There are specific chromosomes which get boosted up to 8-fold their coverage, and those play a key role in the evolution of macroscopic size, cellular elongation, and big size.
That's a lot.
There's a lot happening.
It's fascinating. My head's exploding with these things, and it just reminds me of Orgel's law number 2 attributed to Francis Crick, which is evolution is smarter than you are.
I love that. Oh, I love that. I have to steal that because that's so true.
It's not me. It was Leslie Orgel said it, and it's attributed to Francis Crick. So I'm always going to say I don't want to call it Orgel's rule. I want to call it Crick's rule. But it makes a lot of sense. And this is really. I also think we don't understand selection the way we think we do. Am I correct in assuming that you could then put further types of changes in stressors or changes in parameters that would lead to further types of speciations with your snowflake yeasts?
Absolutely. I mentioned earlier us putting, sperm whale myoglobin into an ancestral snowflake yeast and then evolving them. And that's one great example of something that we can do. Another thing we've done that's really cool is put a rhodopsin, which is a small proton pump that a lot of bacteria and archaea use as a way of generating just a little bit of supplemental ATP, a supplemental it's
Hmm.
a little solar panel that complements heterotrophic
Hmm.
metabolism. And we took that from the fungus which makes a corn smut, yeast metis. We put it into our yeast, and it localizes to the vacuole, where it actually provides energy. They get an energy benefit, so they become, you know, phototrophic. So we're putting both of these into snowflake yeast and seeing how this changes their evolution. That's one great example. Another one is, we haven't done this, but we want to, is taking our yeast that act like little volcanoes and pump fluids through their tissues very quickly, and evolving them with that essentially as a static organism, kind of like a sea sponge, where they have to flow food for the flow media for their energy, for their food, and see how that changes the evolution of morphology. Right? I think that would be a very cool experiment. Right now, they're just being tumbled and mixed constantly, so they don't really get to use that behavior very much. But I would love to evolve them with the use of that behavior. So that's another great example of changing the environment and asking, how does that change their evolution? And there's many more experiments that we could do, but those are the three that we've thought the most about.
Do you think that multicellularity is going to be found elsewhere in the universe?
Um, yeah, I mean, just caveat that I'm basically just kind of like guessing, right? We don't know, but the constraints on multicellularity aren't super high. If you
Mhm.
have cells, it's trivially easy for cells to form groups of cells. Like any single celled organism you have, I bet you can find a way to make it to evolve it to form groups. Like that part's really easy. Then after that, what we've seen with snowflake yeast is that it's really not that hard to get the kinds of shift in Darwinian dynamics that allow groups to become units of selection and units of adaptation, right? The kinds of, if you get simple branching, breaking that branching divides genetic variation into different groups. Selection can act on emergent multicellular traits and drive open-ended, adaptation in those in multicellular traits. Um, None of it to me seems highly constrained. It is an interesting question. I don't think it's yet answered. Why, you know, cyanobacteria were evolving multicellularity over 3 billion years ago. We've had multicellularity on Earth from basically about as long as we've had cells. And, we're evidentiary limited more than I think, we have great evidence cyanobacteria existed over 3 billion years ago, and we're multicellular. My guess is that we had other multicellular bacteria basically going up to the origin of cells, but we just don't have the data. But nothing really evolved the classic eukaryotic, large, spatially explicit, complex multicellularity until the last billion years. And then that evolved in a bunch of different lineages. We have hypotheses for why that's the case, but we don't really know what the answer there is. And so to answer your question, I think of another planet. If you had planetary scales of time, of ecology, of diverse organisms, there are many times that forming groups of cells would be adaptive. And these early benefits of multicellularity, survival, motility, collective metabolism, those benefits would favor the evolution of simple multicellularity. And then you have a bit of an open question of, under what conditions do simple things become complex, right? And what are the things that held it back? What are the things that catalyze it? We have a recent paper on this in PNAS from earlier this year that I think provides a pretty nice candidate answer to that. But it would take, again, a little while to dive into that and explain it. But there are a number of hypotheses that can explain. Some of them are geological in nature, sort of like the oxygenation of Earth happening, really getting modern levels of oxygen about 1 billion years ago, 700 million years ago, sort of really
Right.
ramping up about 1 billion years ago. Other people have argued that the snowball Earth and great cooling that we had between 720 and 630 billion years ago increased the viscosity of the oceans, favoring the evolution of larger, mobile, heterotrophic multicellular organisms. There's a lot of different explanations. My explanation has nothing to do with either of those. But there's lots of candidate explanations. And I think until we kind of resolve that, like, why did it take so long for complex multicellularity to rise on Earth? I don't think we can answer the astrobiology question. Simple
No,
multicellular life, for sure. Early, it's going to happen. I would expect it would happen early, and it would happen basically every time. But
I
things
think
like plants, animals, fungi, and seaweeds, that's harder.
I think that we always have to come back to Jeff Goldblum's line from
Mm,
Jurassic Park. Life will find a way, but life has to start at least.
that's
And
true.
that's what I hope we'll find.
Yes, agreed. I mean, if you had, something as a bacteria, I would be blown away because that's already solved almost all the great challenges of life, right? If you have an information storage system and you have a ribosome to do something with it, and you have lipids to create a membrane, and you have a rotary ATPase to turn chemical gradients into proton gradients into chemical currents, a common chemical currency. If you have those few things, you've already got all the really hard stuff done, I think.
Will, this has been just a fantastic episode. I hope
Thank
that
you.
we can have you back to talk more about this.
It'd be a
You are
pleasure.
like you're like a supernova of ideas. I haven't even asked you about your farm at home with all the
Yeah,
critters.
yeah, yeah,
You
yeah.
have all these great things in your life. You're it's like you're a super juggler. It's like you have developed all these extra arms similar to your snowflake yeasts that enable you to keep all these things in the air. And
Well, I think the secret to that is honestly, I get to work with extremely talented people every day,
yeah.
and it's really due to the community, right? We have
Mhm.
sort of a super organism in the
Yes.
scientific community of people that are super competent, super smart, very collaborative, and helpful to each other. And so to the extent that I get to be a spokesperson for the group, that's wonderful. But it reflects a vibrant community that I'm so happy to be a part of.
I also want to tell listeners and viewers that there are two articles that involve the work of Will, and they're from the science magazine Quanta, and I'm going
Yeah.
to include links to those in the show notes, as well as anything else that Will would like you to see,
Cool.
incidentally, in the show notes. And I really, really want to thank you for your time. It's been such a privilege to have you on episode number 50.
Nifty 50. Thank you so much, Mark. It's been great being here.
Thank you
I
so
look forward
much.
to meeting you in person one day soon.
Be good. Bye
Cool.
now.
Bye.
This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode, with truly wonderful links, can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department at the University of Puget Sound in Tacoma, Washington. Dr. Will Ratcliff is professor and co-director of interdisciplinary PhD in quantitative bioscience at the Georgia Institute of Technology in Atlanta, Georgia. Many thanks to David Renata for superb editing, and Reber Clark for the wonderfully quirky music that makes me smile every time I hear it. I hope that you've all enjoyed being part of our quality quorum for today. See you next time on Matters Microbial.